Hello. Hello, everyone who is here and who will watch the dead stream, and welcome to the Roach Wiggle, December 15th, 2022. We have a stream that will probably, wow, that's interesting. There's a disparity in time between my phone and my computer. This will probably be a average, slightly shorter than average live stream. I have company arriving at 8, so it will be somewhat tentative dependent on that when the live stream will wrap up. I assure you all that we will have a pleasant, cozy, long live stream next Thursday, as next Thursday will be my sabbatical week. And actually, I need to go ahead and update the percentage. There are a couple more donations have come in the last, or at least since I last updated it. Sorry, it's one of those, looking at my inbox and seeing that there was a message that said like urgent due, and I thought, oh boy, what did I forget to pay now? And it was just some thing like urgent due, your application for this sale or something. Very predatory. So yes, next week, hello Tennyson and Jojo, anybody else who's hiding in the shadows today. We boy, I can delete the auction, all the auction documents, because today, and I will preach about this later too, today the last auction order went out. Chris's order went out today. It took me since Monday, many hours of packing every day to get everything. Hello, A. I don't think I've seen you before. So yeah, so the Chris's order went out today. 4-ish days of packing, multiple hours a day. I was doing two major things at the same time. We had the millipede enclosure refreshing, which I have been putting off doing since August or September. I think I brought that up, and packing Chris's order. So I was multitasking those two things. It's kind of a nightmare. The bug room is kind of a nightmare right now. There's substrate all over the floor and tanks and stuff in places that they usually are not. I'm moving shelves around. I'm reorganizing all of this not so fun stuff. I will be getting a good chunk of other things done tomorrow. But wow, I can get rid of these 8 documents that have been sitting on my desktop since October. I can finally delete them all now that everybody's gotten their things. Wow, it's a kind of empty desktop. But anyways, so yes, I'm going to go right ahead, tally up our donations, and see where we're at. I don't think that we are at the goal, but we don't have to be, because I have already committed myself to doing the sabbatical next week, and I will just leave the donate button up until we hit it and then take it down, and that'll be that. And if there's a need to do another one of these, like for an isopod one, if I don't end up getting to that, then I'll put it up again. All right, we are 90% of the way to funding the care guide. So that's amazing. I think I put it up the end of November. Must have been the dead end of November, and the goal is within grasp for the cockroach care guide. Maybe I'll do another one again in the future if I decide to finally do the isopod care guide. I would like to do some YouTube videos on things. He says, yeah, I'm not in the right time zone, so it's my first time catching live. I come from Russ's following. Oh, well, welcome. And Jojo, more donations can always come in. You're very generous. I appreciate everything that everybody has done, big or small amounts. They all add up. So I'm going to go ahead and I'm going to go ahead and update this right now that we are 90% of the way. And yeah, so there we are. And Chris, if you watch this afterwards or you're watching this now, you didn't even have to donate anything because everybody else pulled a Chris and did all the donating and did all the everything. So yeah, it's been great. Could not have asked for better with this, and we're going to be there next week. 5 days of me doing work on the care guide. I was thinking maybe I would double task or multitask and pull up the old care guide, maybe make some notes on where, you know what, this is actually something I feel like I should do with hard copies. And actually I need to replace my printer ink cartridges. So I will probably do that Monday next week. I'll go through, figure out my plan of attack, what I'd like to add. And yeah, and by the end of the week, everybody who donated will receive a copy to proofread, to look at, to make some recommendations for things. And hopefully then what I would do is release it as soon as I can. So let's see, by the end of the week, that's Friday. So I'll probably release it after Christmas. Monday and Tuesday that week are going to be pretty heavy shipping days. But maybe by the live stream of the week of the 26th, I will release it. Crap, I can't do it the 29th because it's going to... So it'll come out within maybe like 2 weeks of being worked on, maybe sooner. I mean, I could submit it to everybody and there could be there may not be any sizable criticisms or desires for things to be added. But speaking of that, this is a stream where I was going to take recommendations, and I'll do this next week too. I was going to take recommendations for or requests for what people would like to see on a bug room tour. So if you were in the chat right now, I'm gonna write something about it. Please submit your requests for the bug room tour, which is coming up. Bug room tour video, which is coming up soon. Submit your requests in the chat. All right. So I put that there. We'll see if chat moves at any tangible pace tonight. If it doesn't, I won't repost it. But if you're here now and you want to see something, ask. Tell me, how do you keep this? How do you do that? How do you whatever? Request it now, and I will write it down in a document. And in addition to the things that I am really wanting to show off in the bug room, I will go through, and for all the requests, I will go through and then further in depth go and look at those things and say, Jojo Macro from the live stream requested to see my Paraplaneta setup. Tennyson Kingsley requested to see my Blabber species Honduras setup. A dot from the live stream requested to see something. So submit those in there. I will start compiling a list. Parcablata Americana setup is number 1 for you. So Jojo Macro, Parcablata, CF Americana set up. All right. So I've got, there we go. There's the first thing on the list. We'll take a look at the setup that I use for the Twain Hearts. And there we go. We got something we can do. I don't know if I'll necessarily go in the order that they're requested. We'll see how many people are requesting things, etc. Oh boy. Yeah, I'll look at my Marulinellas from Marulinella setups. I did get babies out of tricolors, but I don't know how well the culture is going to do over time. But I do have a colony of, I guess they're not Marulinellas anymore, but they were the species Vietnams. Junior would like to see Uricotis and Procera. All right. Also, welcome, Junior. And of course, you're always welcome to ask questions here. A. says, I have 2 generations already, but same wing anxious for how it evolves over time. Yeah, it's something that I'm going to work on and continue to dump time and energy into, is figuring out Marulinellas in a way that works for me, because I have a lot of other things that are really thriving, like Troglodillos and Cubaris, and sometimes just figuring out what the thing is. Phyllosia muscorum. I sent out a group of those that captive bred that Chris won in the auction, but I didn't have any babies in the enclosure right now. And they do, Phyllosia muscorum does have, they recorded under lab settings, they have like 2 reproductive events or phases where when they're half grown they may reproduce, and then when they reach a full size they'll reproduce. But it's not necessarily it's not something it's like in the wild it's temperature related, but in captivity they just sort of like end up being those 2 stages throughout the life of the colony. Hello, some eggs. Good to see you too. I have it in the chat right now to submit requests for things to see on the bug room tour, which should be something. Updated the homepage to reach that the funding for the guide is currently at 90%. I'm curious about other things that I would like to update the guide with. I need to send an email asking the person who sent me these megaphasma if he recommends diapause. All right, submitted that. I'm going to put these megaphasma eggs back on the shelf for now. I think last week, large feeder steps like master feeder, roach containers, or mealworms. All right, some eggs. Would like to see some eggs, large feeder enclosures. Braden would like to see the A. gigas enclosure. Braden, A. gigas. All right. I did actually start finding quite a few baby gigas, and not as many as the first time that I had adults when I first got them and raised a bunch of them, but enough to make me optimistic. And I have made a couple available. There are some sales pending on some quantities of them, but I, on the flip side of things, may have been because I didn't water recently or haven't been paying the best of attention to the adults, but I did lose my my only adult male. Seems to be a common occurrence with people who have gigas, is they'll lose their males before their females, and then they'll be like, oh, I'm not going to be able to find another male. So, but he's been, I think he was 2. It doesn't seem like that's too long for them to live, or it doesn't seem like it seems like that's an awfully short for them. So again, it may have been my husbandry recently, or he could have just been sexed out like it happens. Joshua Campos would like to see Guyana, Cappuccina, and Bisanulata enclosures. Okay. But yeah, so then I redid my millipedes. I had pulled like, I don't know, maybe like 8 to 10 gigas as they had slowly popped up initially. And when I checked on them with my lack of millipede husbandry, or just something that I've noticed gigas do, I'm down to 2 of those. They both look fine, but I'm debating whether or not I want to raise a bunch of gigas singly. I just have so many. I've historically had so many problems with them apparently eating each other, damaging each other. Junior wants to see troglodylos. So yeah, it's a work in progress. I'm trying to. Tennyson wants to see ectobia enclosures, so I'll probably get to anyways. Yeah, it's a problem that I've been trying to and longing to get an entire shelf of Gigas going. Yeah, I can't necessarily justify that at the moment, but I do have a lot of containers. I've been moving adults between containers. I've forced myself to pull babies when I find them now. But I wish I could figure out exactly what was going on. And another person that has had. All right, I can work with that, Tennyson. You want to see Balta Naculata, Balta Latiblatella, Obopterella, and Uricotus. All right. But yeah, it's so this the person that I had sold some to when I first had the gush of babies, she has been getting babies and has had a similar phenomenon hatching. I went and visited her in the spring to help split up Tennyson Senian enclosures. I visited her in the spring to help her check through the setup and split some things up, and there were not as many babies as I hoped, but she did already have some surprisingly that were very far along in development, some offspring that she'd produced. So actually I figured I could ask her for a male if I needed one, I guess. So we're getting them to propagate enough for a couple of sales here and there. Not quite what I would hope for with something like this, where I want whatever happened initially, where I had like hundreds of babies, and for some time it was like that. But yeah, it seems cannibalism of babies eating other babies is an issue, or adults eating babies. Adults eating babies. Statement definitely don't want taken out of context. But yeah, so I do have a couple of containers of them. I have a general plan of attack. I'm just hoping that moving into the future, I can have a group of them always, so that if I keep throwing husbandry at the wall, something will stick and I'll be able to figure out what it is they need. They do seem to like more nutritious food than other millipedes. Like a lot of things will be very a lot of millipedes I've worked with will be very content with just wood or leaves or whatever their thing is. But the gigas definitely seem to like a little bit more than that, like the produce and flakes and whatnot. Somebody asked if I would ever get into apiculture. Yes, it's actually illegal in the city of Livonia. I do have a neighbor who does it, though. It's one of those things where it's like having it's on the books that you can't propagate bees, but there's nothing on the books about having roosters, which is it's very selective enforcement, obviously, with all this stuff. But Yeah, so I would like to do it eventually. Not now. I want more. I need more space and whatnot, but I will get into it eventually. Chris, why are you starting to question your viewing choices? Is it because you've made some questionable decisions in the last couple of months about living thing acquisitions? So yeah, let's see anything else that's going on, because this is a very busy time. This is my first winter in a while. Oh, you're enjoying winter time about adults eating children. Yeah, I'm trying to figure it out. And another thorn in my side. I'm finding the polycarb lids are perfect for me because they're I can bump humidity in enclosures. A lot of my millipede problems were coming from the material that I'm using when it dries out because I'm lazy and I don't want to water the millipedes for some reason. When it dries out, it kind of mats up and forms an impenetrable chunk of dry material that the millipedes don't know or can't burrow into like they can with moist material. They're basically designed to burrow, pretty much all millipedes. And this is it's a saving grace for some things, and it's a bane for other things, like so stuff like Chicoblis. It sucks because they're very dry tolerant, so at least a lot of those survive. They stay on the top of the material and sort of cluster up under other stuff. So it sucks for them, but other things obviously don't do so well when things dry out. I think the only millipedes that I lost, my Thai blues are not doing well. I'm going to end up getting more of those from Oren, I think. I need to split up some Tanzanian earthworms and some albino Narcissus americanus. Although a lot of the other Narcissus are doing fine. But it is, again, on the flip side of things, that layer produces, it's basically like a lid within the enclosure, a gigantic piece of wood. So it does trap humidity of different substrate material beneath it. And some of the millipedes are content to just come up and graze on the bottom of it or in the matrix and stuff below it. So I did produce some Aphiloria or Sigmoria from the ones that I collected in the spring in Huntsville in an enclosure with this stuff happening. And so, and urids do amazing in those setups because they have this perfect humid microclimate beneath the sort of hardened wood up top, and they do amazing. I have from maybe 3 to 5 different localities, and at least 2 different genre of, at least both the genre, I guess, of the urids from the U.S., and they're doing very well for me. So that's very exciting for me. I love urids, easy flat millipedes. But again, some stuff like it was a Cladocricus, Cladocricus, not doing so well. Surprisingly, my flame legs are doing very well that I got from Oren last year in February. They're doing very well. It's a very slow growing strain compared to the one that I had in like 2012 or something like that. But the flame legs are doing phenomenal. I have a colony of those tuberillos, or whatever the proper Latin is for them. The Borneo alligator isopods. There's a good colony of them in with the flame legs. Very easy. Really like doing combo setups when I can. And so yeah, there's been some good. There's been some bad of millipedes. I lost my big pine keys. Fortunately, Kai gave me some more that he and Alan had collected, and he had produced some. So those, I have more, but obviously not going to have any available for a while. Archibald and conservation and whatnot do have available, or will have available. Some stuff like Anodinobolus ocraceus just infested their containers and are doing exceptional. And so I need to add millipedes to the site. I really need to. But Leptogoniulus, Benoitulus, binoculus. So I have a couple of things going that are doing well. A couple of things I wish were doing better. Something Epibolus pulchripes. That's another one that I have. I think I have 1 male and 3 females, or 2 males and 3 females, or something like that. Excited to get those going again. Those are very easy species. My Onomerus that I collected in Alabama, I took some of the wood that I found them in, and the worst thing that could happen, there was a stone centipede in with them, so wiped out all of the Onomerus. And so I'm upset about that. Hopefully Alan will show me that location again this year when I go to visit in spring, and I'll collect another starter set, and I will not give them their substrate material that I found them with. But yeah, that sucked. But a lot of the others, again, there's some highs and some lows, and in general, rocking that like 70% success rate, and I'm okay with that. Just a shame the millipedes are kind of very expensive. At least the niftiest ones are. So Dilophoraptor, good to see you. I'm going to go back through chat and touch base with everybody that I have ignored. Dilophoraptor, good to see you again. I think it's been a while. Some eggs ask, Kyle, do you have any mammal projects that are of interest right now? At the expo last Saturday, I picked up a pair of African dwarf dormice, Graphiurus murinus, and they were cute. Like, Kai, why would you eat that? That's so cute. Why wouldn't you want that? That's so cute. Kai actually did not find them cute. I think that they're very adorable. So I picked up a pair of them. They love insects. That's primarily why I got them, is I looked up like the lab care guide. Some of these lab care guides just crack me up. So you read a husbandry guide that they make for these, and it's saying things like, yes, we raise them on this cottage cheese and maple syrup and this and this. And they're suggesting, how to take care of them in the laboratory. And then while you're going through their diet discoveries, when they did the testing for, dietary preferences, and they talk about all that stuff, like the weird stuff like the cottage cheese and the maple syrup and things like that. Then you see that there's like a little footnote about like, over all things, they preferred mealworms and insect prey or whatever. But then when they go and give the I think it was maybe even the abstract where they recommend what to feed them in the lab, I don't think they made it a point to say insects. And then all the stuff in the wild diet, it says like insects. So it's like, feed them insects. It's not difficult to raise a colony of some feeder insect in captivity, and yet you recommend all this other stuff. It's actually more difficult and more expensive to go out and buy cottage cheese and maple syrup than it is to raise a colony of mealworms or lobster roaches and feed that to them in situ. So I don't know. It's just one of those funny things where they kind of not really contradict themselves, but it's just an interesting. They don't really. I know that they don't want people to worry about having to maintain them to raise insect colonies, but again, it's actually not that difficult. And they've been ravenous feeding on pre-killed stuff that I've given them. They love it, and I'm wondering if I'm going to go on a tangent here. I'm wondering if they'll be very easy to breed for me because of I'm offering them insect prey. I had a friend in high school who really liked birds, and for a short period of time he had a bunch of Christmas money, and we were just sort of like I was trying to spread the love of raising animals to all the people that I know, and he had an interest in birds. I was like, well, why don't you get into breeding birds? You love birds, work with some birds and try to breed them, and you're feeling unsatisfied and unmotivated, and like you have no goal in existence. So here, try something that will give you a very finite goal. And so he worked with, one of the things that he bought was a pair of cutthroat finches, which all the information we had read at the time was that, it's a very difficult species to breed. They always kill their babies. They always do this. And they made some footnotes about like, yes, give them some dried mealworms or something. So being friends with the person who had bugs, I was able to give him live bugs, mealworms and roaches and whatnot. And he ended up breeding the cutthroat finches like it was nothing. I think that he lost one of the offspring, or no, he let them raise like a couple of the offspring, but one of them got pushed out of the nest, so he ended up hand raising it. And he had a hand raised cutthroat finch. And it was just, so interesting. But it's funny how in these other hobbies, same thing with fish keeping, where they talk about like, oh, you must raise live food for them and stuff like this. It's like, yeah, like the stimulation of eating stuff, the nutrient profile of eating something that they naturally eat and it's digestible in a very natural way to, yeah, you're gonna be able to breed things you didn't think you'd be able to breed if you feed them a better diet. And doing that on your, the grindle worms and banana worms and vinegar eels and all that fun stuff. So yeah, it's just like this is like the things that eat bugs, feed the bugs, So anyway, some eggs, there's my tangent. And yeah, so now I'm working with these. We'll see how they end up doing over time, if at all. So hopefully I'll just keep feeding them roaches and we'll see how they do. And maybe one day babies will pop up and that'll be exciting. Tennyson asks, also some of your Vulgari projects, specifically the high yellow. Oh, you wanted to see some of those. Oh, yes, the Novas. So Nova, we figured out, is actually a disease, a pathogen, a virus that causes a disease in crustaceans that is called marble disease. Was it marble? Porcelain disease. Porcelain disease. Which is caused by I can just see what this is. But what sort of organism is it? I'm trying to figure out. Microsporidian parasite, protozoan parasite. So yes, the Nova vulgare they have porcelain disease. No, porcelain. Is it porcelain? What did I just say? Goldfish brain. Yes, porcelain disease. But interestingly, I think this may also be what causes the mixture of phenotypes in the California or Cali mix Porcellio levis, and what has caused a lot of the color variation in Porcellio hoffmannsegii as well as species Seville. And it pops up in other things occasionally too, including Porcellio dilatatus. I think that's what causes the lighter phenotype that sometimes pops up in Porcellio dilatatus as well. Because again, trying to establish cultures of those, they lead to nothing. It's likely that the Porcellio levis cultivar, the Cali mix, has some degree of resistance to it, which is why you're able to keep colonies going. Because some individuals are going to die because they're fully susceptible to it. Some of them have some sort of naturally gained resistance. So Jojo asked for Narcissus americanus, which that's going to be exciting. We'll look at a container of just wood. It's going to be very thrilling. Just sawdust, basically. Let's see. Dorleo orani and Blabberus giganteus would be cool to see as well. All right. I hear the boy knocking at the back door, but he is outside eating ribs right now. Chris says, I knew alligators could be hybridized with isopods at the Tuberillo alligator isopods. Very cool, stupidly tiny. I haven't really gotten into all the spiny micropods and stuff like that. I can't justify it to myself. Maybe after I get tricolors going, I can justify something like that. I do want pseudo armadillo sexpinosus. I had those. They did very well for me. Not the seller's fault, but I ended up with all males. you get a bad roll sometimes. That's just all you can say and do sometimes. So I would like pseudo armadillos again. I'm actually digging some of the obscure large species of Porcellio that I've cobbled together over the last year. Porcellio wagneri being one of those, and a little bit more recently, I think it was around the same time, maybe not. Porcellio species, Almeria Mountains, and those have been pretty nifty for me. So I've been enjoying the Porcellios, and it seems like a lot of people have shifted away from those, unfortunately. Junior would like to. Do they make a good friend at the? They haven't tried to bite me. The African dormice, they haven't tried to bite me. They seem to be very sugar glidery in behavior. I think that hand raising or interacting with the babies will make them much more friendly, and so that's just something I'm gonna do if they have babies. Again, this is kind of borderline impulse buy. Thought they were cute, could justify it. I do like my small mammals, so we'll see. Junior wants to see Bantua robusta. Which, I mean, we could even see Bantua robusta tonight. Even it's just right up there. It's one of those small containers. Tennyson asked about the Montgomery pieds. Those are more promising. I put a set of them in that I pulled out of the main colony in with some Blabrus colossius. And I'd recently gotten perused back from Joshua Campos. Thank you for saving those, Joshua, if you're still here. I put a small group of them in with those. The Colossius grew very rapidly and kind of stressed them out, and all of the Armadillidium vulgari died. So, anyways, I set up a container with just the pieds from that line, and they had babies. They're still alive. So fingers crossed. We'll see. It should prove out pretty quickly. It seems simple, recessive. Again, it's very heritable. That colony was started from a single pair that I put into that container. And now there's hundreds, very durable line. I got to start releasing my isopod projects. I've been holding a lot of stuff back. Tennyson wants to see the Hoffman Zegii and Pulchellum. Be interesting your big communal you have. Chain asks, are your Narcissus reclusive? Mine seem mainly arboreal. Mine can be pretty reclusive, but also with the well ventilated setups, usually they come out after a water. And now that I'm trying these polycarb lids, there a lot of things are staying out in about 24/7, which is really cool, especially with the millipedes. But I've run into a problem. What's that? What is this over here? I've run into a problem in that the polycarb company that I got the first set of lids from, which I'm in love with, is out of material to make them, and so they contacted me and said, do you want a refund or do you want to wait? And I said, I'll wait. Like, I need these. Like, I need them. And so I had to budget what polycarb lids I gave to what millipedes, and kind of stressful because now I have to water a couple of them more frequently than the others, which kind of sucks. Let's see. Can you put them on your shoulder, Nausicaa style? I don't know. They seem. I don't know if they were when I came into this room. They're currently in the room over there because it's. I had read that they may go into a I guess it's a bromation or a diapause of some sort, where you call it for mammals when it's not actually hibernation. If the temperature is in the high 60s to I thought, oh, I'll just keep them in the office where it's always pretty warm. But when I came in last night, they kind of like freaked out and hid. But when I picked them up, they're very docile during the day. I don't know if it's just because they're tired or I've woken them up. Jose Nunez says, hello, Sam, missed part of the stream, but in time for more bugs. Yes, hello and welcome. I don't know if I think I've seen you maybe once or twice before, Jose. It's easy to pick out regulars. I can say that much. Tennyson, you're asking for, there's a lot of things you want to see, Tennyson, and some of these things will be on the list. I will do my best, but I have to try and accommodate everybody, although it will probably be a longer video. Crickets in general will be cool to see. I'll put that down for you, Chris. Crickets are something I would like to continue expanding the number of Grylus that I have. I did get. Gotta make sure that they're hydrated. Yeah, I did get babies from the massive Grylus that Satchel sent me. I'm paranoid about losing them to entomophagus fungus or whatever, which I did lose a couple of things from the Arizona trip after I got a generation to fungi and weird care and whatever. So, but I decided I would pull these until they get to like third instar and then move them into a final enclosure because I really do not want to lose these giant Grylus. Taphiliska. Chris says, speaking of insectivores, anything that eats insects would be cool. Someone's been building feeder colonies without anything to feed them to. Or some random small rodents, the zebra mice. They used to be those African pygmy mice. Reminds me, I need to get in touch with that rodent person from Ohio. See if African pygmy mice or Santa Rosa mice. A lot of those things are insectivorous, and that's probably why people have some ups and downs with them in captivity. Some exist. Do you have any roach planted tanks? Not really. A lot of my tanks they're geared entirely towards the roaches, and I do find the aesthetic of just a tank with a bunch of roaches in it very appealing. I don't necessarily feel the need to add a plant to it. There's also the risk of if you get a plant from somewhere that it may have a Pycnoscelis turnamensis in it. Those are very common potted plants, even from like the store or at different vendors. Braden would like to see any communal setup with many species. So that'd probably be a mixture of roaches and whatnot. Currently in this room, there's a communal of the Princessia, Androhamana, new strain of Armadillium maculatum I'm calling bubblegum, Horselia hasi, high yellows, and Dipteroptrum hanstromi. And so that's one. Some of those things I'll pull out as I get the numbers that I want, and they will eventually hit an equilibrium. But in general, it's very easy to keep one roach with one isopod. Paraplaneta missionary said hello and wants to see a Paraplaneta showcase. I'll do that. I'll go. We'll look at some Paraplaneta. Actually, I had some very interesting adults pop up in the mile zero culture lately since we're getting into a couple generations in. And the original pair, I think it was the male that had a very interesting pronotum marking. And so I sent a picture to Alan because there was one that popped up looking very, very similar to Paraplaneta australacei. And I was like, is this a hybrid? I know in captivity they've kept cultures of Americana and Australacei and Bernea together for years and not have any hybrids pop up. And Alan said that this phenotype had already been documented in Americana. So it may just be a coalescence of similar genes. But I pulled the female, and I'm going to see if it's strongly heritable and what I can do with it. I have a feeling it is. It'll make things very confusing, I guess, if I don't give it a good strain name. I'm the guy that you sent out to pester Will about about pseudoscorpions. Yeah, Will's supposed to come over on Sunday. I know he's been very busy with existence lately, since he's not over here slaving away in the Roach mines this year. But I know he's been very busy. Rough green snakes. Yeah, I have rough green snakes. I've been told that they'll eat gyna species. Mine, I just feed crickets too. They seem to like them the most, but there's videos of them eating all kinds of stuff like dragonflies. Smooth green snakes, which I think are what's in our area in the Midwest, I think are a little pickier, but I wouldn't be surprised if they ate roaches. It's the place where I saw some. There were also Paraplaneta Parcoblata pennsylvanica, but probably things like caterpillars and crickets and grasshoppers. Temperature and lighting can also play a big role in whether or not snakes will eat some sorts of snakes. So, yeah, I'm trying to think if there's anything else that I wanted to touch on today. We've looked at the donations. Again, I'll bring it up since we got a lot of people in here. The donations were at 90% for the husbandry guide. I would anticipate at the rate the funding's been going that we'll be at 100% shortly, which is wonderful. Jojo asks, do you have any frogs? I have some spadefoot toads that I collected over the summer on the Arizona trip. And Brandon keeps his amphibians here. Primarily, I gave him a spadefoot toad that I collected. He has some cane toads. He has some White's tree frogs. He had a tomato frog that I think I got him for Christmas last year, but it pushed out of the enclosure and desiccated, which is unfortunate. So I do like frogs. I like greenhouse frogs a lot too. I figure that one day when I have a gigantic Florida greenhouse thing, one of my life goals is to have a big old Florida style greenhouse thing where it's in the high 70s every month of the year and tropical foliage and all that fun stuff. So it's a life goal, but I figure I will pick some sort of amphibian that I really like. Obviously something tropical to release into there and hopefully get a population going in some capacity. It's just a matter of finding something that's cool enough that I could justify releasing into this micro ecosystem that I'd be starting. I used to have a couple of fire skinks, but 4 or 5 years ago, I really got out of herps. I couldn't justify heating the extra room downstairs to keep my herp collection going when I was going through my rough times. And now that was actually a good idea because now I have a cool or cold room that I can put stuff that needs to diapause in. Speaking of which, I need to pull the Parcoblata caudali out of there to see if they will, after 2 or so years, if they'll all start to mature to adulthood instead of doing stationary molts. I also threw a bunch of dynasties in there for the winter. So you should show us your Masola as well. More people need to see those. We got a couple minutes here. I think I'm going to wrap up the stream after this, but since Tennyson asked, and we do need, I should show Bug tonight. Let's see some Masola histers. I've got my first babies that I produced have been maturing, and let's take a look at some of them because they're really cool. And they will be very popular over time. Oh, come on, stop being so hissy. Stop being so hissy. Got a mix of sizes here. Not quite the array of coloration and whatnot that I wanted to show off, but we have a very beautiful subadult male here. Okay, put the hand up front and try to block my ugly mug to get the focus on the bug. What they say? Put your hand. There we go. See if we can get a good focus here on this guy eventually. There we go. All right. It's got a subadult off my middle finger here. You can see either a springtail or a mite crawling by my callus. Let's just ignore that. And we have, whoops, this, remember, they're excellent climbers, everybody. Oh, everything's going to hell in a handbasket. I did not like a disruption of the status quo there. The female's chilling on my other hand, but I want to show off this male. I'll show off this male here. Ah. Can you guys hear him? He's probably picking up the hissing as he's having his little hissy fit. Yeah, so we got this lovely, let me try this again. This lovely adult male here. I should focus eventually. There we go. Lovely adult male here. There's been a really good diversity of stripe extent in this, the individuals that I've seen. And I've debated whether or not I want to get the numbers up before I pulled some for selective breeding. And TJ sent me a couple of random unrelated individuals. And so I decided, you know what, I will pull a couple because I'm not worried about inbreeding. As you guys know, I don't worry about inbreeding with bugs 99% of the time. But I would like to preserve the genetic diversity of this strain before I go breeding the crap out of them. And so I want to make sure that I get a good outcrossing of individuals in the starting stock for this colony. So here's a nice adult male, got some abdominal striping. And some of them have striping all over the abdomen too, but they still have this very rich orange or reddish color. Let's look at an adult female. Maybe the first time that I've ever seen a Gronfodorina jump. This male just jumped off of my hand onto the keyboard. First time. So here we have a Masola female. You can see there's some striping on the abdomen there. You don't really see this in the Cleveland strain, and not even so much in the other strain that I have, which is the UCRs. Don't really seem to see this that much. I'm trying. Handful of hissers. Shove the thick abdomen into the camera. Oh yeah. I should focus again eventually. Just want to show off that striping, and I need it to focus to do that. I'll get a camera with manual focus in the future. Maybe this camera can do that. I don't know. Back there, that's a little better. Anyways, so you can see some of the striping there. There's some females that have a very rich red abdomen, and this female is kind of bordering on that. There's a couple of females that have more of the tiger striping too. So yeah, I really enjoy this strain. I am kind of a sucker for the browns and whatnot in the Cleveland strain. And of course, you out of that came the mahoganies and some of the other projects that I'm working with. But these are really cool. They've got very high contrast. Um, I again am a horrible person and kind of interested in how these genetics can be selectively bred for different traits, but simultaneously, it's just nice to have a different line of Portentosa going around and stock that has kind of a locality. I think I've talked about that before. They have a locality name, but in Madagascar they refer to like 20 places as like the same name, and so we don't know exactly where what Masola in Madagascar the strain name refers to. The information has been lost, but we at least know that it is pure stock straight out of Madagascar and has been maintained in labs and institutions in Europe. So but yeah, very exciting year for hissing cockroaches. Lots of species available. Lots of projects coming ready. Things like that. It has been a very good year for hissers. If only TJ and I could get these freaking Andromana Princessia to have babies, then it would be the icing on the cake. But they're just obstinate. They just don't want to have babies. The Portentosa from Masola are already going to have another generation. These Princessia have not produced anything. I'll go through here and read some comments. Everybody's showbug. A couple micro geckos Chris Snyder has tried. They require just as much or more work as regular geckos. Viper geckos, etc. TJ trying to separate some high red Masolas. I think that could be worthwhile to get a nice red strain. Yeah, so I have a couple project ideas. And again, I can kind of justify it because TJ did send me a couple, like all those individuals were siblings that you just saw from one pairing. TJ only sent me one pair of Masolas. And so I think I see Brandon pulling up. And so I wanted to outbreed to preserve the genetic diversity present in this strain as best as I could. And so maybe not. So I have the couple that he sent me that are unrelated, this first pair and their offspring. I'm going to co-house those all, in pairs for a bit, or put them all into one enclosure so that I get outbreeding, and then eventually I will, so there's a couple litters that are outbred, and then eventually I will collapse everything in together to just get a little bit more diversity in there and hopefully keep it over time. Chain, I don't care about inbreeding because in my experience, it does not seem to affect captive invertebrates in a tangible and meaningful way. Far more important is making sure that your husbandry, if you start to encounter problems, that your husbandry is identical to what you were doing before, on the order of, am I using the same wood pellets? Do I have the same humidity? Do I have the same enclosure? Are they at the same density that they were when I started with them? Are they in the same physical condition? Is their gut microbiome the same? Have they been stressed? Have I moved? Have they moved? Are more important questions to ask, because usually with respect to inbreeding, those things get filtered out of the gene pool very quickly after they're in captivity. For example, if you had a simple recessive gene that was lethal for homozygous recessives, you would see that your first generation, would normally be like, let's say it's like a hisser or something, 40 babies. You would find that 25% of them die in the first generation. And the next generation, you see a slightly less ratio than that dying, because that allele is not going to persist that long in the population over time. It's a different story if you have only 4 individuals. But again, if you're assuming that inbreeding is imbuing some sort of terrible detrimental trait to something, you're going to see purifying selection within the first couple of generations as the population adapts to captivity, versus some people will have a big colony of roaches going for years. You got mixed life stages, mixed generations, etc., and all of a sudden the colony starts to collapse and they say, oh, they were inbred. That's not how that works. You don't suddenly have all these different life stages, collapse. You don't have a colony collapse due to inbreeding in such a sudden period of time. It's a gradual process that happens with each iteration of generations, if it happens at all. The only insects that I believe in captivity, in my opinion, that are severely affected by inbreeding are things that have very hardwired mechanisms against siblings reproducing for whatever reason. One of those, I feel, are orchid mantids, because you have adult males maturing months and months and months before females will of the same cohort, if they're kept under the same conditions. And there do seem to be infertility problems in pairing siblings for some reason. But again, that's my personal experience with things with roaches. I don't worry about it at all. With isopods, I don't worry about it at all. Basically, people would be shocked if they knew how few individuals some of our strains of isopods and roaches and other insects have been started with. The best example being Elliptorhina davidi was started with one adult female. And for years, I struggled with them and thought, I would do well. And then I would struggle and I'd say, inbreeding, must be inbreeding, however. No, I didn't have their husbandry right. They wanted to be baked alive is what they wanted. And now it's a very reliable species for me. And I know I'll never lose that species. So yeah, you got to look at the husbandry. You got to look at what dissect what you are doing differently or what conditions are different. Again, putting 10 invertebrates into an enclosure and seeing them do well and reproduce, and suddenly you've got 100 in that same space. Well, now you've added another variable that you have to take into account. Are they eating food faster? Are they bumping into each other and stressing each other out more frequently? Are they producing something in their waste that is accumulating faster and not breaking down fast enough now that there's 100 of them in that space instead of 10? And so those are the things that I point a finger at with colonies over time, basically. But yeah, it's all perspective, because inbreeding is a natural function of natural selection. It happens all the time with island adapted organisms. There are vertebrate examples where inbreeding did not cause any sort of meaningful, did not cause any long-term impact on the populations. Sure, not being as heterozygous inherently means that a population is not as capable of adapting to new conditions, which is what people usually mean by inbreeding is bad, is that the population is not able to adapt over time because it simply doesn't have the diversity of characteristics to adapt to new conditions over time. But there is nothing intrinsically wrong with it, at least in a lot of organisms. You look at things like the Mauritius kestrel, which was down to a population of about 3, I think, at some point. And the population itself was likely never very large to begin with. It's a bird that lives on the island of Mauritius, and the population, due to volcanic activity, due to all kinds of other ecological factors, has probably fluctuated down into the single digits historically, before humans were involved. And so population went to 3 due to some new influence, like, lack of conservation measures and introduced species, etc. And then due to conservation efforts, which then produced more individuals, the population, I believe, now fluctuates somewhere between 50 and 100 birds. Last time I had checked. I'm going to look this up really quick to make sure I don't sound like an idiot. By 1974, the Mauritius kestrel was close to extinction, with only 4 known birds, including 1 breeding female. After a number of conservation efforts, by 2019, there was a population of about 400 birds. This conservation achievement is regarded as one of the most successful and best documented bird restoration projects in the world. And just some quick notes to add to that as context. Apparently, in pre-colonial times, the population was estimated between 175 and 325 breeding pairs. And then there's the stating about things, the introduction of various invasive species and deforestation, environmental changes caused that to happen. So, just doing a little bit of reading to catch up on things. Some apparent inbreeding depression was noted in the captive birds. It was certainly lower than might be expected, given that the affected population size is maybe 5 and blah blah blah. So they note in here the cumulative effects of DDT and environmental changes were a much larger factor than that. So, but anyway, so there's a real world example of not just a captive colony, but a species that is now derived from extremely inbred stock in wild situations that has recovered to a point of being relatively sustainable from a conservation perspective. And again, Tennyson, yeah, I see you asked about separating pairs for outbreeding. I am not doing this because I believe it will affect the health of the stock. I am doing it to try and preserve the genetic diversity present in the stock, in case it is of importance at some point. Either, I remember we had these really cool looking ones that popped up. They used to have these color markings and now they don't pop up anymore, or what have you. Because preserving genetic diversity is important, but I will not fret about inbreeding in colonies because of some perception about it being intrinsically bad for their health or whatever. So some eggs notes that inbreeding occurs in populations of humans, such as in the state of Alabama. I think Alan can attest to that. Lower Alabama rules. Kyle is trying really hard to justify inbreeding. Somebody check his browser history. It is important to note that I am an only child and always have been. So anybody who checks my browser history should find that I have a clean conscience. West Virginia for the win. More good people. So, but it happened in humans, obviously. Not that we're necessarily more complex, but you have a different phenotypic presentation of things that matter in humans. In society, it matters to be a functioning human being. It just so happens that apparently a lot of diseases present from recessive alleles, or a lot of mental disabilities you can point a finger at recessive alleles, or even at one of the more well-studied physiological examples being sickle cell anemia, where it is advantageous to be heterozygous for that in certain parts of Africa because it conveys resistance to malaria, versus homozygous recessive, where you have a debilitating condition. And I don't think I ever really read up on whether or not people who are double homozygous recessive for that allele that causes sickle cell anemia, if they live long, if they're functional in life. I don't think I ever looked that up or was informed of that. But anyway, but here's the other thing. If it were environmentally advantageous, and we have to use the broadest term for environmentally here, in the sense that on a planetary scale, if it were advantageous for humans to be 10 IQ, eating dirt, and playing League of Legends all day, then the population would become stupider over time, because there's a selective pressure. For whatever reason, these individuals that would have 10 IQ would be able to reproduce more. And ultimately, from an evolutionary perspective, what matters is whether or not traits are advantageous and whether or not they persist over time, and whether or not you reproduce enough. And so if there were selective pressure, should society ever turn to that, or society collapse or whatever, sure, people will get stupider. It doesn't matter. There's no, if it's beneficial from a reproductive perspective and from a resource acquisition perspective to become a paranoid schizophrenic, it's going to be selected for. That's just how it is. I mean, maybe some of those traits explain behaviors in other organisms and other mammals. So again, it's all about the context of the situation from an environmental and chronological perspective. And but obviously it was more advantageous for us to have holistic mental health, or at least for a period of time, than to be paranoid schizophrenics. And with that, I will end this week's Roach Wiggle. I hear Brandon knocking on the front door. So I'll see everybody next week in the middle of the Roach Care Guide work. I hope everybody has a wonderful week until then. And for especially this applies to Chris, may your cultures ever be bountiful. Take care, everybody, and I will see you next week.